From Micro TV, this is TWiEVO, This Week in Evolution, episode 114, recorded on the 21st, actually it's live streamed on the 21st of May, 2025. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Utah, Nels LD.
Hey, Vincent, great to be with you for the 114th time. We're we're a little stumbling out of the blocks here a little bit, updating our links and getting our audio connections and video connections all worked out, but we're here. And we're glad to be with you all who are here as well. Thank you for your patience. We're up in live streaming as close as I can tell.
We had some technical glitches, but we solved them, and we are ready to dispense some science. Let me welcome our moderators today. Tom from Western Oregon and Barb Mack from the UK. Tell us where you're from. Thanks for moderating, folks. And tell us where you're from. We'd love to shout you out. I see here we have Elizabeth. Where is Elizabeth? I forgot. But John is in Eden Prairie, Minnesota, which Nels knows, right?
Know quite well. Watching with interest. There's actually a big basketball series going on between the Minnesota Timberwolves and the Oklahoma City Thunder. And the Thunder happened to have a player with some Minnesota background who actually went to my old high school, a guy called Chet Holmgren, who plays in the NBA. He's doing quite well. So anyway, the series started in Oklahoma City, but we'll be moving to Minneapolis soon.
So Gary says, Denver sunny and 63, perfect for growing peas. Why would you say that? Who knows, Nels, right?
Oh, wow. We're foreshadowing here a little bit of our science that we'll be discussing today on TWiEVO. Welcome. Glad to have you here.
Vanity's here, another moderator. Thank you, Vanity, for joining us. Glad you could make it. I know you're very busy.
Fantastic.
John says, the Thunder is very good, and hopefully we will match up better in the series.
Yeah, we'll see. Down 0-1. The first game was last night, and Timberwolves were looking good. Or maybe the Thunder just took a while to warm up. But anyway, we'll see what happens. A lot of basketball left to play there.
Anybody else want to say hi? We're open for business. We have
open for business.
Science and science. We have good information here. By the way, if you enjoy these programs, we'd love to have your support. As you notice, we don't do ads on any of our programs. The way we support these programs is through listener donations. So we depend on you because there are production costs associated with this, of course. Yeah. And your donations help us do it. So go to microbe.tv/contribute to find the different ways that you can help us out. So we have some new people here. FSM is the dog. Missed seeing live for a while. We usually do once a month, right, Nels?
Yeah. Usually towards the end of the month, third week, Wednesday, 1 p.m. Eastern time in the U.S. And great to have everyone along when you can make it. And the fun part about live streaming is we're on the lookout in the chat stream to hear from you, hear some of your questions or comments, in addition to saying hello. So thank you all for being here. And
FSM is in Wales, where it's 17C.
Great.
And wet.
Yeah, it's been chilly here, wet and chilly here, but we'll take it.
Yeah, it's also wet here. Vanity is freezing on Long Island, 56F, and rainy. It's funny, freezing, 56 is not really freezing, but
that's right.
It feels chilly. I mean, it was a little bit warm a week ago, and now, boom, rain and chill.
A lot of ups and downs. I was reading yesterday that it's massively unseasonably warm in Greenland and Iceland. and that's sort of the imbalance here. So the entire, as we think about the whole Earth, more warmth than cold, but the U.S. is definitely sort of in a trough at the moment. But
Bird Carver is in Massachusetts. It's quite a name, Bird Carver.
I like it. Yeah, great to have you along.
Vanity always likes looking at your whiteboard.
Yeah,
They
it's been
can't.
better focused right now. It's a mess.
Does it change now? Do you use it?
I do, just in one corner, mostly in the lower corner, and then the rest of it is almost just sort of a pattern. But it's been, I look at it from time to time, and it is real science, so it is things that we're working on. If anyone can squeeze any meaning or value out of the scribbles behind me, I would be great. I would be grateful to know. But I'm not worried about getting scooped or something. It's mostly, pretty detailed sort of scribblings from the top of the head. Yeah.
John writes, I always donate at the evening stream. So tonight we have a live stream, office hours, 8 p.m.
Awesome.
Eastern.
Thank you.
Want Nels to know it's meant to support all the great programming, including TWiEVO.
Yeah, I really appreciate that. And so, we're we're doing this not to make a lot of money or any money, but to keep the conversations going, to keep the lines of communication open. And so, really appreciate the support, not just in person as we have these conversations, but the resources too, so that we can keep our connections live, keep the infrastructure of micro TV going. So thank you all.
Bird says it's too cold for tomatoes.
I know it. I'm trying to get some starts in the ground here. I've got something like 50 pepper plants, about 24 tomato starts, and I'm pausing to try to get a little warmer weather to get things going here, including in Salt Lake.
You don't get warm. It's just,
Oh yeah, it's on the way. And then by the time we're talking on TWiEVO 115, it'll probably be, too hot and dry. So it's all over the map, as they say.
So everybody's looking at your board, and Tom says, look like some polyadenylation there, the string of A.
Yep. Guilty as charged. We've so one of our, I won't go into the details here, but some poly-A's in a poxvirus genome in some of our projects that are coming along. We've used that to diagnose how these viruses gain genes from their hosts. And so,
So the pox transcripts are weird because they have poly-A at the 5' end, right?
Yeah, exactly. Both 5' and 3'. Some really interesting genetic gymnastics by the pox viruses that we're still their labs actively working to understand. Yeah, it's really cool.
So Bird Carver points out Novavax will be available. That's right, 65 and over. And other people, if you have some issues, maybe this. I don't think it's great, but the people in charge are in charge. They get to do what they want.
Oh my goodness. Well, that will. I think we'll sprinkle some. Have some as we'll get to the paper sooner than later here. But I think as we go, Vincent, I have a few points that I want to raise about sort of current events as it relates to science, the practice of science going forward here.
Heart and Soul is from Ethiopia, or an Ethiopian. Welcome, Heart and Soul.
Yeah,
thank you.
To have
Glad
you
like our souls shows. And SFSM says dummies making dumb rules.
Oof.
Agree.
It's getting more wild as we go.
Agree.
Yep.
Dummies making dumb rules, and then they try to justify them with BS.
That's right. Yeah, it's wild out there. We've been talking about it as a Category 4 hurricane against science here. Feels like the winds are picking up even in the last week or two. Some of the attacks on science and some of the legislation that's coming through. I think some things to be really concerned about and to discuss and put on the radar here if it isn't already kind of coming through the news headlines.
Yep. Steen, welcome, Steen. Steen is a plant virologist. I will reveal that because I know Steen.
Awesome.
The FDA leadership seems to have gotten much more active on YouTube. That's right. Makari has released some videos of his propaganda, but you could go look at them and see what they have to say. The fact is that over 200 kids have died of COVID.
Mm-hmm.
Now, you may say, oh, it's a small number, but you know what? If it's your kid,
Mm.
you're pretty upset that you couldn't prevent it. So this is just nonsense, considering, the very low risk of any kind of damage from the vaccine. So I don't see why people can't say, I want it and have it, right? Because right now, if you have this recommendation and you don't have any illness, you will not be able to get this vaccine and it won't be paid for by your health insurance. So,
Yeah. Well, and that goes directly back on RFK Jr. I mean, you can find it's not hard to find him going back on his word or reversing it
yeah,
completely. But that was one of the things he said in his confirmation hearing was that they weren't going to take approved vaccines away, and here we are. And so this notion of make your own choice, it's between you and your doctor. We flipped the script, and a lot of those small government conservatives are looking pretty big government and reaching in and
yep.
making the decisions for us at the moment. Wow, pretty garbled, pretty scrambled in terms of.
FSM. I found myself agreeing with RFK. Don't take medical advice from me.
That's right. That's the one point where we can all find some common ground here for sure. Yikes! It's getting
So
weirder as we go.
John wants to know if pregnant women will still be able to. No, actually, which is bad because COVID in pregnant women is bad. Exactly. They can't get it, as far as I remember. Adults 65 and older. And if you're 12 to 64, you have to have at least one health problem that puts you at increased risk. Is pregnancy a health problem that puts you at increased risk? I don't think it's a problem, right? But it does put you at increased risk.
Indeed.
And Peter asks, is MAGA evolving into Christo-fascism? Yeah, for sure. But I don't know. The conservative Christians, I don't think they're religious at all. It's just the guys to do crappy things to the U.S., to take over, basically, to take over the democracy. Because they think only certain people should have the power. And, right now, up until January, the power was distributed in the U.S. pretty well. And they're just chipping away at it. I don't know, Nels, it's very frustrating, as you know.
Oh, yeah, of course. Yeah, and I mean, I don't think the notion. So, a lot of religions, world religions, Christianity, etc., being sort of twisted or used to justify things that's not sort of new to since January. That's been going on for all of human history to some degree,
of course,
or post civilization. But this strain of it is pretty troubling and sort of feels especially sort of hypocritical, like that. Just the the expression of whatever mega related Christianity. You don't have to go too far. For example, the new Pope, who is from Chicago originally, the first American derived Pope. There are some great memes coming up related to that, but he pointed out before he was appointed how some of the statements that J.D. Vance was making about sort of you should love your close knit family the most, and then sort of less like a hierarchical sort of love structure. The Pope was pretty strong on social media saying, actually, no, that's not how Christianity works. So it's been, it's definitely been pretzeled or garbled or put into the use by people in ways that are, I think, don't do much justice to sort of the deeper meaning of these religious traditions. There's no question about that.
Well, FSM says those at the bottom are religious, exploited by those at the top who aren't.
Yeah, it's an interesting point. Yeah, some clear trend lines showing up. We're also seeing that exploitation potentially showing up in the new big budget bill, big government bill that's being thrown around in Washington, D.C. right now. I think I was just before that we picked up on the podcast looking at some of those budget numbers and the tax sort of cuts going to the rich at the expense of the poor and the services that we rely on as a country, sort of the social contract being chipped away or worse if this thing goes through. Interesting to see what happens the next few weeks here as that process continues to unfold.
The main objective, Nels, is to cut programs to save money so you can give tax breaks to corporations and wealthy individuals. That's it.
Yeah, those numbers are becoming more and more clear as the CBO, the Congressional Budget Office, is starting to put that into focus. What this really looks like from the
Steve is in Hawaii, Kauai. How they
welcome.
sleep at night. They don't care. They don't care,
Yeah, yeah.
or they're too dumb to care.
Or
I mean,
actually, they do
did you
sleep at
see?
night. So, so the
There was a Senate hearing,
yeah, yeah.
and they asked the lady, I can't remember, no, Christy Nome, right? They asked her,
Oh,
what's
habeas corpus.
habeas corpus? And she got it all wrong. She didn't even know what it was.
Yeah, it's sort of a
She
key
said it's the ability of the president to do what he wants with people.
to
No.
deport people. Yeah,
Well, she said, well, he can't. No, you can't. You have Congress only has that right. She doesn't know anything. This is so typical. Anyway, too much politics.
yeah. But actually, they are sleeping at night. So there is a in part of the budget markup. There's a local interest story here. So actually, my representative in Congress, a fellow called Blake Moore. So they're doing a lot of the markup stuff in the middle of the night,
Mm-hmm.
and he was caught on camera sleeping through a lot of it. So not only are they sleeping at night, but they're sleeping while they're trying to conduct the business that they're not proud enough to do during the day because it's so backwards in terms of the distribution of resources here across the board. The slash, I mean, incredible slashing of the investments in science going forward. It's Category 4 hurricane time for not just science, or not just for the whole country, but for science as well. And so,
So Gary
hoping,
says it was Category 2 hit in 1981, especially for EPA and Interior.
yeah. It's a good analogy. The analogy works in a lot of ways. It's not perfect, but for me, it gets at some of what we're up against.
M0, welcome. Good to have you.
Good to be here.
Vanity says
Good to have you
Christy
here.
Nome is a puppy killer. I can't get past that.
Yeah, this is not the leadership here. The notion that this is the greatest cabinet ever,
Oh,
almost by the hour, seems
oh
to
my
get
gosh.
gets
It's the
weak.
worst.
Yeah,
Completely unqualified. And
yeah.
people who do not even read and understand what they're talking about. Like Christine Nome, she didn't know what habeas corpus was. She's never read it.
Yeah, and
That's
then
ridiculous.
the folks that do have more experience or background in some of these areas, Marco Rubio, for example, has flipped the script on what he's talking about. But Vincent, this if this podcast were called "This Week in Politics," we could keep running for
Yeah, we'll
another.
stop now. Yeah. But
We'll
here's
get
one
back.
more. One more.
Okay,
Cabinet
please.
by IKEA. That's about it. Yeah.
I saw that one. Or you see that sign at some of the protests, some of the Stand Up for Science protests, for example. I've seen better cabinets at IKEA.
Yep.
I think that's a good one. That connects. Okay, so we'll keep sprinkling in some politics here, especially as it relates to the practice of science in the U.S., which is all intertwined with the practice of science around the world, all of the international sort of connections that are made. And we'll see that in our paper today too. So we're not going to be talking about vaccines. We're not going to be talking about humans. We're going to be talking about plants and some, a topic of very deep interest to geneticists, which is the pea plants that Mendel worked on and the genetic basis of the traits that came forward 160 years ago or so, 160 plus years ago, Mendelian inheritance, the principles of heredity.
Here we go.
Yeah, this is fun. So the paper, Genomic and Genetic Insights into Mendel's Pea Genes. And before we dive right in, let's talk about who did the work. So 5 co-first authors, Kong Feng, Baizi Chen, Julie Hofer, Yan Shi, and Mi Zhang. Senior authors, I think there's 3 senior authors, sort of co-corresponding authors. This is Noel Ellis, Noam Chayut, and Shifeng Cheng. So as with a lot of the science, almost all of the science that we talk about on TWiEVO, international ties here. This one coming mostly out of China. So the Agricultural Genomics Institute in Shenzhen. This is in the Chinese Academy of Agricultural Sciences in Shenzhen, China. Also, the John Innes Center, so Norwich Research Park in Norwich, UK, which I think has a lot of a massive collection of pea plants that's traced back to Mendel's time. And so a key contribution there, the materials that we'll be talking about, the pea plants and the genomes of them that are analyzed in the work. Also, some connections with laboratories, Institute of Plant Sciences in Paris, Paleogenomics Laboratory, also in France, and the EMBL, European Molecular Biology Lab, the Wellcome Genome Campus in Hixton, United Kingdom. Minnesota is back in the headlines here, Vincent. So the Department of Plant Pathology, University of Minnesota. I think one of the authors is who worked on this project and moved there, is my impression. And then also Beijing, China. So the state key laboratory of weed improvement. Again, international team. So when we look at the funding here, actually, this is one of the rare papers where I did not find an acknowledgement of National Institutes of Health funding, a lot of which is in danger at the moment, given the budget scenario. But that's pretty unusual. Most of our papers have at least 1, 2, or 3 grants, somewhere in that ballpark, NIH. Although, if we stay on our current trajectory, I would say that probably a lot more of the research that we consider will have less NIH funding going
This
forward. So
probably is long before the cuts, but
long before the cuts, but that's like we're looking at the trend lines here. And what position does the U.S. want to have in the sort of the scientific conversation as we potentially take away a lot of our competitive advantage, our the resources that we put in here, our seat at the table for really impactful, interesting work going forward, all in jeopardy at the moment. Okay, so let's move back to the science here, right? And I think for me, Vincent, this is like catnip topic for TWiEVO, where we take sort of this historical science topic, Mendel's peas, this foundational. We all learned this in probably middle school, high school, college biology. We'll unpack that or try to brush off some of the dust on what Mendel's experiments were and what they meant, why they were so important. And then the reason this is sort of a catnip topic for TWiEVO is that it now takes these genomic tools that are possible, the ability to just sequence the bejesus out of all of these genomes, in this case, the pea plants, and then to ask questions about how that genetics ties to the traits, the phenotypes, or the sort of iconic traits of the pea plants that were used to deduce the principles of heredity. And the authors here, frame the study here, trace this back 160 years ago to the anniversary of Mendel's lectures on the inheritance of traits in peas, and describe this as the best experiments ever done. What do you think, Vincent? Do you buy that?
I thought your experiments were the best.
Well, you never know, right? So I guess it's published a week ago, so
they
or
were
a few weeks ago.
probably among the first very well done experiments, right? Mm-hmm. And people have accused him of fraud, and we'll get back to that at the end of this.
Yeah, exactly.
But I mean, it was quite interesting. I mean, I guess he was interested in improving plants, right, for crops. So that's why he did this. And at the time, we didn't know what a gene was. We knew about inheritance, right? We knew about natural selection and evolution, but we didn't know what it was acting on. We didn't know about DNA, of course, right?
Of course.
And so Mendel said there's something discrete in the genome that passes on traits. Very important.
Very important foundational stuff that's held up for 160, like Mendelian genetics is still a thing. And so what were these experiments? He went to the seed catalogs that already existed. So there had been a lot of what we've called
was it Burpee? Burpee?
I don't know if it was Burpee. It might be that. I think some of these companies go way back. And so, so it had all of these sort of distinct traits, right? Seed color, yellow or green, pod color, or we'll get into the famous characteristics, wrinkled seeds or smooth seeds. Really good insight here that those distinctions by, in those traits that came out through the breeding, the selective breeding, or as we've discussed on TWiEVO before, this idea of conscious versus unconscious selection. So when humans have consciously tried to get phenotypes, have done this selective breeding, you end up with sometimes called artificial selection. I really like that. Conscious selection is a better. We discussed this back on TWiEVO episode 96, which was what was that one called? It's something going bananas over the origins of maize or the origins of corn. That notion came up, and so here these experiments were quite simple, right? So to take the seeds, grow up the plants, and then do crosses of plants that showed those different like charismatic traits, whether it's the flower color or location, etc. Um, and then as he collected the offspring, the F1s and the F2s, so the first generation from the offspring of the parents, the next generation, the offspring of those pea plants, and then just by following those traits, able to deduce before we knew the basis of DNA or genes, as you pointed out, Vincent, how this works. So he ended up deriving three principles of heredity. They're probably familiar to us: the law of dominance. So this is versus the recessive or masked traits, right? Do you need two copies of a gene? So Mendel coined the idea of an allele, of a difference or a variant in the gene. That the law of segregation, you get one allele from each parent,
Mm.
and that's how those patterns came out of those crosses. The law of independent assortment, as you look between two traits, not just one, that as long as they're not linked on the same chromosome, which again, sort of added a little cloudiness way back 160 years ago, that these traits could move independently of each other if they weren't linked. And so, yeah, I think a fair idea that these could have been the best experiments of all time in terms of the lasting impact on
Yes.
our knowledge of
Well,
genetics.
sure. If that's what the bar is for a great experiment, yeah, it's had incredible impact on genetics, right?
Yeah, and held up the notion
Punnett
of all.
squares, remember
There
those?
you go. Exactly. That's how we first learned, and even probably felt cloudy to a lot of us, myself included. As I was training early as a geneticist, it gets complicated in a hurry. And in fact, I think this paper, or if we now come forward 160 years, use all of the sort of powerful tools that are possible with genomics and genetic analysis. Still a very clear, endorsement of those principles, but also starts to show the sort of complicated details that go into the phenotypes as it relates to the genotypes here. And so this wasn't totally, a shot out of the dark here. So of Mendel's sort of 7 famous P traits that we'll march through in a moment here, 4 have already been described at the genetic level. And so they already had sort of a foothold here. And I think this is important to mention, Vincent, to just pause and think about, all of the science we do really is just sort of how kind of the connective tissue of science. All of the work before, it's really rare to get a complete breakthrough. All of the science we do really depends on the work of others and building on it.
Mm.
And this is an example of that. So 4 of these traits have already been described. They sort of go into more detail or learn new things about that. But for the other 3, less was known. So actually, it's been proposed what the genes are that underlie some of the other traits, but it hadn't really been nailed down. And so that's sort of marks the advance of the work, or the impact of this work, as it probably really gives strong evidence that these are the sort of genomic, the genetic variation that's at play for the other 3 of Mendel's iconic traits. And then, so we'll talk about the details here in a moment, but once they've sort of used genomic tools to really go deep on some of the diversity of the pea plants, sort of those famous Mendel peas and the sort of close relatives of them, they also extend their work to 70 other traits that are sort of commonly thought of as important for plant breeding strategies into the future. And so this helps to start to give some clues about the molecular basis for some of these things. How can you sort of influence the traits to get outcomes in agricultural applications that might mean better yields, more resistance to, pests, et cetera, things like that. Okay, but to really, we're TWiEVO. We're not This Week in Genetics either. Twig, we're TWiEVO. And so I think we want to put this in the bigger context, which is where does it all start with these domesticated pea plants? And so here the cultivation was in the Fertile Crescent, we estimate something like 10,000 years ago. And then that's when sort of we can trace the beginnings of humans getting into the mix here, that conscious selection, where breeders, have this intuition. If I collect the the seeds from these plants, grow them, cross them with these ones, maybe I'll get a better feature, and sort of the selective breeding to improve crops, to get the characteristics that make them more palatable, more productive, more profitable in those ways. So the 7 traits that Mendel sort of jumped in on 160 years ago were traits that are involved in seed shape. Was the R locus, or the R region, or R gene. Plant height, which we called LE. Seed color, which was I, and flower color, which was A. The three, and so other scientists have convincingly shown the genes that are probably at play, or the variation in the genes that are at play, that will influence the seed shape, the plant height, the seed color, or the flower color. The other 3, pod form, are proposed to be 2 loci, the P and the V genetic regions. Pod color, which is known as GP by Mendel. And then the 7th one, the last one, fascication, or elongated growth of flowers, or the flower location, sometimes referred to as cresting. I'm not a plant geneticist or plant biologist, but I looked this up, as you might expect, late last night on Wikipedia. Some of the coolest fascication phenotypes are in saguaro cactuses, where they're like growing up, and then there's this like crest that shows up near the top. And there's sort of, a genetic variation that goes into that. For the pea plants, I think this relates to the location of the flowers and how the stems sort of develop. So there can be sort of on one end of this trait or phenotype is the bifurcation of stems. And then on the other sort of continuum of this phenotype is a cluster of flowers out sort of at the apex of the plant there. So anyway, that's kind of getting into the details. I think the bigger, if we step back and sort of think about the big message here, are these really distinct morphological traits that Mendel very cleverly chose in order to get at the genetic basis. I think some luck at play here, but also some real intuition. So, flower color, right? If that's our eyes, like pick that up very clearly. It's a discrete trait where you can say it's either this color or that color, with little bit, in this case, for these crosses, with a little variation in between. So anyway, so that's sort of my sort of awkward attempt at a little bit of the history of Mendel's genetics. So then what did these authors do? So they put together a diversity panel, what they call a diversity panel of pea plants. And so this relies on the seed collections that are out there, getting samples so that they can get genomic DNA. And then they collected in all several species in the Pisum genus, which includes the cultivated pea plants that we're all familiar with, and probably many of us have grown ourselves. And then, so some wild, some domesticated, and they get 700 samples here, where they do short read sequencing to the tune of about 80 gigabases of DNA sequence. Just, this is actually, in terms of sort of the technology here, I don't think this is sort of like out of scale. The human genome is 3.3 gigabases, remember. But what this allows them to do is get roughly 20x coverage of the genomes of all of their 700 samples. And from that, in sort of the early analysis, they measure, catalog 150 million genetic differences between these samples. And so part of kind of behind the scenes here, and thinking about this through the TWiEVO lens, is as an evolutionary biologist, evolutionary geneticist, you really want to make kind of careful choices here about the tree length. So how related or distantly related are these samples? And the hope here is that they're closely enough related that the genetic changes that underlie those phenotypic changes, what's the color of the flower, is the seed coat wrinkled or smooth, that the genetic changes will be in a common ancestor, that they'll all be the same. They'll be selected consciously over time by plant breeders. But then as you go back to sift through the genomes and try to connect the genotypes, the genetic differences, to the phenotypes, the trait differences that you see with your eyes, is it all from the same source? If you pick 2 distantly related samples, what could happen is it's actually really interesting biology, evolutionary biology, but they could converge. There could be separate, independent sort of, changes in the genome that lead to the same traits. So here, to get cleaner signal, they're trying to put together, a panel of ones that are close enough related that it comes the signal or the association between genotype and phenotype is all from the same source. So they build a phylogenetic tree. So you can take these sequences. I actually don't remember offhand which specific sequences they used to make the tree, but they make the point that the tree doesn't look like sort of the classic tree of life sort of thing. It's more of a bush. It's all kind of the branches are in some cases are low confidence. How are they most related to each other? And that reflects the human intervention, all of the breeding, right? So there's been a lot of introgression or crosses. And so something that seems more wild was brought in at some point. And so there's all this sort of complicated or entangled history of genetics among the
Take a
700
look. Here, show it to you here.
Yeah, there it is. So
on the right.
So this is kind of a high level look at the 700 samples. On the right is that phylogenetic bush, let's call it. And then the color coding gets at, the different species or sort of isolates that they're using. And then to the left there, all of these bars, that gets at the 150, this is a way of kind of graphically trying to summarize chromosome to chromosome, the level of genetic diversity there, which gets to the 150 million genetic differences that they map. Okay, so that kind of sets the table in terms of the genomic analysis. And then from there, they take all of this, sort of new information or this diversity panel and try to apply that to the 7 traits, traits, and get to the genetic basis of that using the new data that they have. And as we said, 4 had already known genes, but then they actually see some new stuff as they go closer. So let's we
So
won't
basically,
belabor it.
Nels, they're
Yep,
doing genome-wide association studies, GWAS, with
correct.
the traits that they're interested in. They're trying to find uh, changes, polymorphisms in the genomes that would associate with them, right?
Exactly. And so the way this works, just at a high level, is like let's take round versus wrinkled seeds. And so in all of your samples across those 700, you'll then say, okay, these ones of the 700, these ones have round seeds.
Mm.
These ones have wrinkled seeds. And then going back to all of the genetic variation, are there variants that track with that round seed versus the wrinkled seed that you can sort of pull out across the whole panel or the majority of the panel, where they share a genetic difference that tracks with that phenotypic difference, the difference in this case for sort of the quality of the seed. Is it round or is it wrinkled? And what they do then is use statistics to propose an association between that genotype, the phenotype, the trait, to the genotype, the genetic variation. And when you do this for the R gene involved in the seed coats, the signal, the statistical signal maps to a gene called SBE1, which is a gene that encodes an enzyme that is involved in the quality of the starch that is put into the seed. So a starch branching enzyme.
Who
And
knew?
well, this
Who
was
knew?
known,
No, but
but I
who
think
knew starch
oh,
could have different
Oh yeah,
qualities, right?
right. Yeah, oh, absolutely. Yeah. So how you sort of process starch could then lead to this difference, right, in the
Yep.
surface of your seed. Yeah. Um, and so, what is the variation between the two iconic traits? And what they found is an IPS-R element, a transposable element in exon 22 of the SBE1 gene. So here we are immediately on the first sort of attack, colliding the genetic changes with transposable elements, selfish genetic elements. And I love this because it kind of ties to the history as well. When we think about Barbara McClintock, the jumping genes that she
Mm.
made famous in corn based on that iconic phenotype of the kernel colors of corn. And so here, different genetic element, but the same theme again and again. And what's wild here, Vincent, right, is that so these transposable elements, there's no sort of intelligent designer here or hand of God. This is basically just a selfish genetic element making a mistake, jumping into as it's sort of propagating, jumping into an exon, happens to have the impact which changes the seed quality. Some breeder thought, oh, that was kind of cool, and then this gets propagated and goes forward sort of as a consequence of conscious selection. So that's number one. We're going to go, we won't belabor it, but we'll just kind of go trait by trait. The I gene, letter I, and this is yellow versus green seed color.
Hmm.
This was already mapped to a gene of magnesium, or a gene that encodes enzyme a magnesium D-ketolase gene, which is involved in chlorophyll degradation. So, of course, chlorophyll, some of the green sort of chemical biology here of plants. It was known that that gene existed in that diversity panel. And so what's new from this work is they find two new alleles, two new variants that can cause that yellow versus the green seed color. And guess what? The first one is an LTR retrotransposon. So we have selfish genes messing with this chlorophyll degradation pathway, which can influence the color of the seeds. They also found, so it's not just the transposable elements that are at play here. They also found a deletion of 408 base pair, that 408 letters of DNA that was deleted in the promoter that drives the expression of that gene, which could also explain that difference. And so, now what they're seeing here in these different alleles are, as they kind of look through that panel, is actually different ways that the variation can show up, how you can change the genotype in different ways to get the same phenotype coming out. And so, I think they're kind of at this interesting level of diversity in their panel where they're still seeing the same sort of impact on the same traits as it relates to the same genes, but sort of different ways of getting to that as you sort of sample more, in essence, more time and more space, genetic space, by looking at a big panel. Next up, the A gene. So this is flower color, and is known to be influenced by BHLH transcription factor. So we're back into alphabet soup here. Helix loop, helix something, transcription factor. And so now these are genes that encode proteins that actually regulate the expression of a lot of genes. And in this case, with a focus on anthocyanin pigmentation. And so that transcription factor is sort of a key node of control for flower color. And again, they see several new variants here that hadn't been described before, but sort of underline the changes to this can then have genotypic changes, can have outcomes on the phenotype. Of course, here the flower color, which is a trait that humans have been interested in for a very long time. In addition to sort of the nutrition that you get from plants or from peas, there's also just the beauty of them, right? And so this is sort of a tie back to that part of the selective breeding. Finally, the LE gene. So this is plant height. 5 haplotypes are known. And so haplotypes is a set of shared genetic variants that sort of describe a region of DNA. There are 5 haplotypes. These are sort of multiple changes shared between the 5 groups. They're changes that are in and around the GA3 oxidase 1 gene. And it turns out that of the 5 haplotypes, just one of them ends up, which is a single point mutation in one of the tracks. So a single DNA base pair change that can lead to reduced height, whereas the other 4 have normal height. And so this, I think it just kind of hints at some of the complexity there as we talk about, the notion of like, how did Mendel's numbers, the Mendelian inheritance, the the recessive traits where you get that 25% of the trait coming out, like the numbers were looking pretty good, but the genetics behind this can be a little bit more complicated. And so was it that he really hit those numbers exactly? Did he sort of polish the numbers a little bit? We'll return to that idea a little bit later. But all of these 4 traits so far that have been described fit squarely into Mendelian genetics. And so in terms of the principles, it was all there, even though this was before we knew what genes were, or obviously didn't have the technology to understand what their sequences are or what that meant. Okay, so now let's move to the 3 other traits that up till now, in some cases, had some proposed genes, but wasn't really tied together. Here, the diversity panel, the GWAS, I think provides some evidence that this probably, that we've narrowed down to the genetic basis of the other 3 traits that Mendel tackled. So the first one is GP, which is pod color, seed pod color, P pod. And so there's green color was described by Mendel as capital GP/capital GP, and this should hopefully trigger some Punnett square memories, whereas the yellow pod color was lowercase gp/lowercase gp. So why are there two? It's one allele from each parent. There's the capital letter and the lowercase letters. If you get two lowercase letters, you've got yellow pod colors. So it turns out that all of the yellow potted lines have 100,000 base pair deletion in a region linked to the trait that was proposed. So this is again using gene mapping and GWAS. And here the deletion actually removes a couple of genes, but also a transposable element, an ogre element. The transposable elements are all over the place here. They're kind of the fingerprints of genetic change that lead to consequential variation mediated by the transposable elements. But there's a gene called the CHL-G gene, which is involved in chlorophyll synthesis again. So we're back to chlorophyll, which makes sense when you're looking at the phenotypes, the green or yellow seed pods here, seed pods. So what's interesting about this deletion is it causes some of the neighboring genes to be located closer to each other. And so when transcription happens, which is the templating, so you have the DNA template, RNA polymerase comes in, makes messenger RNA, which is intermediate before the gene code becomes the protein, in this case, a chlorophyll synthesis protein. But because of that deletion, you have these genes that were sort of located in the area are now in closer proximity. The RNA polymerase makes a mistake or has a variation where it fuses transcripts together between several genes. So you get these alternate, so-called splice forms of these genes at the mRNA level. And it kind of messes up the protein, the chlorophyll synthesis protein in those ones that have the yellow pod color. So that's new biology that they sort of traced it to that and proposed that that's it makes sense from what we know about that gene, but they wanted to test that. So they actually make a mutant line that messes up that gene, and lo and behold, you have yellow pod plants. So it seems like they're probably in the right ballpark there, linking the genetic changes to the actual biology or to the trait. Okay, next up is a double locus, P and V. This relates to the pod shape. So, and I think, again, my sort of Mendelian genetics, P genetics is a little rusty here, covered in dust, but the pod shape here sort of plays out as actually the sort of quality of the so-called parchment of the pod. Is it more edible, right? So here the conscious selection is if you have a really tough pod, you're probably not going to eat it. You might still eat the peas or use the peas, right? But the pod itself, when you're just like picking these up and eating them like ripe off the vine, if there's a difference in the parchment, you'd select for that. These are vegetable peas that are easier to eat. So the P gene, they mapped first using genetic GWAS. So they have their massive panel among the 700 samples, 700 genomes, and they connect that to the pod phenotype here. And the ones that have, so they map it to a 8.3 megabase region. That's on chromosome 1. And further sort of narrow that to a high scoring candidate gene. That score is based on sort of statistical association between the trait, the lack of pod parchment, to the gene. So it encodes a dodeca CLE peptide. And here again, I made the point earlier that biology sort of, whether it's evolutionary genetics, all of biology sort of depends on the work of others, right? And so it turns out that gene has been studied, and the protein that it makes is identical or is a relative in Arabidopsis. So this is mouse ear cress, a weed that was made into a genetic model system, Arabidopsis, which has been studied widely by plant biologists, plant geneticists. And there's what's called a tracheary element differentiation inhibitory factor, or TDIF, of Arabidopsis, which maps to this exact, or the equivalent of the same gene, this peptide from the pea plant that they track it to. And Arabidopsis, this regulates the formation of xylem. And so there's a single premature stop codon. And when that happens, when you interrupt that gene, it means the consequence for the plant biology is that you have more xylem, and that messes with the pod shape or the pod parchment, the quality of the pod. So they draw that first connection. And then for the second gene, the V gene, their GWAS mapped it to chromosome 6, so a different chromosome, unlinked chromosome. And they narrow it there to a 2 megabase segment that includes a gene called MYB26. And this is another transcription factor. So you land not just on the sort of genes that make enzymes or proteins, but ones that control sets of genes that then can kind of regulate complex biology. And that regulates, it's known in Arabidopsis, again, sort of tying to what's known about the plant biology here, to pod wall thickening in Arabidopsis. So obviously Arabidopsis doesn't have like the beautiful pea pods that you eat, but it does make pods where it puts all the seeds, and then those get broadcast and spread as the weed spreads. So here they again do some experiments. So they use a technique called virus-induced gene silencing to mess with those genetic regions, the P and the V, and they get consistent results here, where they mess with the pod shape if they mess with those genetic regions. And again, so as you then look across that diversity panel, they see these single letter changes in some cases that they're mapping here, but they also see other ways that they're probably sort of evolution or human genetics has messed with these genes, human genetic interventions, where there's transposons that are hopping around and probably messing with that biology as well. Okay, last one, Vincent. Fasciation or flower position. So here, the FA gene or the FA region. This is a trait that can range from the stem bifurcation all the way to the clustering of flowers. And again, sort of standing on the shoulder of giants, previous work had suggested a gene on chromosome 4. They have their sort of massive data set, and not too surprisingly, as they run that through, they find a 15 megabase region on chromosome 4. They narrow it down further to a gene. This is total alphabet soup. I'm not going to say it, but it's a gene base that's described for its location because it doesn't have a functional annotation yet. This is new biology, right? So this is pretty exciting. And as you get from what's been proposed before from sort of the genetic region to like actually the gene, and having that convincing argument for the high resolution genomic data, they're able to sort of, propose that this is a gene that you'd prioritize and figure out what it's doing because it has this really sort of interesting phenotypic influence or implication. So the gene that co-segregates with fasciation is there's a bunch of haplotypes. Again, it's haplotype 3 of the gene, has a 5 base pair deletion in the second exon of the gene. This causes a frame shift or a premature stop codon. The protein is not functional anymore. And then that's what explains the fasciation in the FA lines that Mendel propagated. Here, we don't know what that gene does. They do try to connect it to a little bit that's known, again, I think from Arabidopsis work, which is just by the a blast search. So you take the sequence of the gene, compare it to other genes that are out there, and ask, is there someone who has studied a related gene that can say something about the function? It looks a little bit like a receptor kinase involved in signaling or biochemical signaling. And this is known in other plants to influence the structure of the shoot apical meristem. So I think that's just sort of this kind of circumstantial evidence that this is probably the right gene to explain that. But it's kind of a fun tour through the detective work of genetics, right? So you use, you don't just sort of start blindly and then just like from whole cloth come up with a completely new discovery. You're using as many sort of orthogonal lines of evidence as you can to try to build the case. And so, this is science, right? We don't immediately get to a truth. We sort of we learn something new, we put it out there, we make comparisons, we see how the work kind of holds together, and then try to advance the field by going that way. So I think we, just in the interest of time, we won't go through the 72 other traits that they that they try to make these genetic links to. But these are, they have this massive data set now, right? And so I think to get our attention, to get our excitement, they link it back to this historical case, Mendel's pea plants, which is really fun to kind of put it together with the history in our own sort of education and science. But maybe the real, kind of advance here by having this diversity panel for peas is then to think about all of the other traits and what's going on with the genetics there in cases where it isn't as well studied. So they highlight 70 plus traits that have agricultural relevance. This includes, other characteristics of the seeds, the size of them, organ size, pods, other characteristics of the flowers, the leaf, the root, plant architecture, characteristics that are all at play among the pea plants. And so the GWAS then, taking all these traits, they point, they have all these signals for the genetic changes, the variants or the alleles that underline those traits. Some are pretty complicated. So these are like quantitative trait loci. So it's like combinations of variation, not just sort of in one haplotype or one region, but actually between genes are at play. And so you might have multiple GWAS peaks on different chromosomes that are mapping. The statistical strength can kind of go up and down. It can get pretty complicated. And associations are not sort of cause and effect. But they do give predictions about what are the genetic changes that might be at play in some of these traits. So why does this all matter? If you understand the basis of these traits and given the sort of tools, the genetic tools to mess with them, this is sort of, conscious selection on steroids instead of the generations and some decades, years of plant breeding that it can take to sort of get traits to segregate in ways that are beneficial. If you can go in and make scalpel-like genetic changes based on this information, you could very quickly get to agricultural crops, whether it's peas, again, or comparing this or other agricultural crops, orchids, whatever it is, if you're interested in like sort of flower morphology, to go in and get at the traits that you want in sort of a more...
Although they point out that still to really do this and to figure out what these changes are doing, they need what they call a mature genetic transformation system and targeted gene editing in pea urgently needed, because there's a lot more work to do, as you point out, and those techniques are needed for that.
Oh, absolutely. Yep, that's right. And so all of the crops, right, are a little bit different. And so different plant geneticists are focusing on tools and different ones. I think the pea has probably been kind of in the background since it's not one of the massive crops that sort of drive a lot of human agriculture, whether it's rice, wheat, or some of the alternatives that are being proposed, sorghum, et cetera. I think there's a lot of investment there currently in the biotechnology to make those scalpel-like changes, less so in peas. And so the work brings the field forward a little bit, but by proposing some of these gene connections, and it might just be that someone working in, let's say, rice or sorghum or something like that, makes a link for a trait that they're excited about, does that search and says, oh, wow, in that diversity panel of peas, this came out too. And that might, in future work sort of say, okay, well, we're probably focusing in the right area here. Let's put some of our limited resources into that space, helps to prioritize the work going forward. And so, this doesn't this isn't the definitive, like all science, this isn't sort of the definitive book on how all of these traits work, but it advances the field a little bit.
I think it's really interesting how this study shows all the different mutational mechanisms that result in these traits, right, that he studied. Point mutations, and these can change the pattern of splicing. They can change the reading frame. They can change the amino acid sequence. They could put stop codons in. You got transposon insertions. You have DNA deletions. You have promoter deletions. You have exon deletions, suppressors. You have the whole genetic toolbox here in these peas. It's amazing.
Now, I love that. And it again underlines, right? Like if you were an engineer starting from scratch and just thinking about how do I design a system, this isn't how you do it. It's a mess, right? It's chaos. It's all of these different mechanisms, stuff jumping around, things getting prematurely truncated. But that's just what worked in that instance. Yeah. And then that's what went forward in the breeding. And so, it's a really fun kind of catalog of the messiness and sloppiness of how genetics works,
So the
whether it's
other
a pea
thing,
or
Nels,
us. Yeah, yeah,
I think is interesting is that they say we really didn't know if GWAS, right, genome-wide association,
yeah.
was powerful enough to do this, right? Because maybe the signal was too weak or too fuzzy. And they
Mhm.
say that peaks are quite strong and they correspond to the expected loci. And they say GWAS is a suitable first step, right? And they say this works really well in peas because most genes are
Correct.
single copy, but it's
Exactly.
not enough. You can't delineate small sets. That's why they use all the other data that you mentioned in this paper.
Exactly.
So I think that's
Yeah.
a very cool validation of GWAS for this species.
Yep. Agreed. And we should be careful though, not to sort of overly hype the praises of GWAS. So for our own
Sure.
species, which doesn't sort of we don't have some of the advantages that you have in this diversity panel of peas, both by luck and by you know the sort of ethical considerations of selective breeding, et cetera, obviously, in our own, breeding history. But yeah, GWAS, the hope, right? So if we can contrast peas with humans, the hope was that the traits, instead of, um, seed color, pod size, et cetera, but things like disease traits, right? So if you
Yep.
take
Yep.
big populations and ask, okay, among these humans that have been diagnosed with this disease, what is the genetic basis of it? By comparing other humans that don't have that, right? And it might
Right.
be within a population, sort of famously, all of the genetics and people in Iceland, et cetera. Decode Genetics is a company that's used this strategy. GWAS has been a mixed bag. So there are certainly a lot of genetic diseases where it's very, like you're saying, it's a single copy gene with a very clear genetic mutation, make the same genetic toolbox as what we see in all of this, truncating mutations, et cetera, transposable elements, the whole 9 yards. But in a lot of cases, and as you get into more complex diseases, things like diabetes, right, or chronic diseases, the genetic basis of that is much more complicated. Contributions from many genes, and that's where the sort of ability of GWAS to draw that connection to genotype to phenotype starts to melt away. And
Right.
so, let's connect this to current events. When RFK Jr. gets up and tries to badger um, Senator Patty Murray that for the last 30 years, we haven't done anything about chronic diseases. Well, the genetics community would say we've done a lot. And one of the things we've learned is that chronic diseases like diabetes are really complicated when you go to the genetics. And so you don't
Mm.
just sort of get to one genetic change or the basis of that and then sort of solve that with a drug. We're still learning how it all fits together, not just the genetic contributions, but the environmental ones. And so it's not that, science
Yeah.
doesn't have
Yeah.
the tools or the ability, it needs the resources these are the frontline things. But when we argue about this or put out these sort of, false notions, we start to drive and take away resources. We start to drive the field backwards. And then we find ourselves in snake oil territory where it's all vitamin A and, take away food dyes, which is not going to solve the problem here. And so this again
Well,
illustrates, I think,
this
this
now
massive disconnect.
this is an uneducated person just saying things that he doesn't know anything about. Right.
Well, he did. He at least had the self awareness to say, "Do not take medical advice from me." I also
Right.
wouldn't take scientific advice from him either, or run all of U.S. science under his auspices because it's.
Nels, I wouldn't even take recreational advice. He went swimming in a stream that's fecal contaminated. Right.
That's right. Yeah, it's wild days,
Nels,
to say
the
the
other
least.
thing I thought was really
Hmm.
we should remark on is
Yeah.
that they say there's some dispute about what Mendel actually did. It's been argued that he was not interested in inheritance or
Right.
that he had a preformed theory of inheritance
Mm-hmm.
that he sought to demonstrate even to the extent of fabricating data. So the
Mm-hmm.
authors say these views are mutually exclusive, of course. You can't
Yeah.
not be interested in inheritance and then fabricate. So they reject it. But also, I would say the data in this paper show he was looking at alleles, and it was their segregation is very clear, right?
Absolutely. No, this is really great. So, they the statement is a little bit vague. They say they reject sort of that there's any, cooking the books here. And I have to confess that I haven't like followed the controversy too closely. I think the things I've heard is that if you go back and just with the number, so when we think about dominance and recessive are the traits that you observe as you count them in the offspring. Is it 1 to 1? Is it 1 to 3, et cetera, in the recessive case? And if you look at those numbers, like they're a little, his numbers hit that pretty strongly. And so did he get it exact? I don't know. But what does hold up, and what's important here is, is it that his numbers are exactly right? And maybe they were, just by it sounds like the chance of that being the case is kind of statistically be like sort of hitting, the lottery or the jackpot or something like that, which is not formally impossible. But what really stands up are the principles here, right? Those 3 principles of heredity: independent assortment,
Yep.
aggregation, and so and I think this is important actually, because science finds itself today under attack in many corners based on this notion of sort of like fraud, cooking the books, etc. Science is a human endeavor practiced by scientists. And I'm not saying so there are clear cases of, fraud, fabrication, etc. But for a lot of science, Mendel included, the concepts have withstood the test of time. 160 years later, this is still useful information as we put it together with the genetics. And so there's a little bit of this, kind of hindsight is 2020 here, where I think the critiques on science don't really match the messiness of that moment of advancing the field. And so if you get the concept right, that can be revolutionary for our understanding of biology. And if some of the details under the sort of human issues, science isn't practiced by robots. If we get distracted by those issues, I think we kind of miss the point or the value here. And so some of the attacks on science are that, well, wait a minute, you got the details wrong here. But if the bigger concept holds, where is the real value here? And this applies, I think, not just to Mendel's peas. It applies to all the science we do. It applies to pandemics, SARS-2. What were the origins of? How do we understand? Like all of these things. And so, as people attack the scientists who were trying to figure out where SARS-2 came from in the very first days, with that sort of hindsight is 2020, this is part of the reason that we're off the rails here. And so I don't have the answer or know what to do, but I want to kind of lift this up or illustrate this issue, which is sort of the details of science, this notion that you get everything 100% correct on the first blush versus the chaos and messiness of it. And how do we better, as scientists, sort of convey that, and that the process is working when we're making mistakes. And in fact, when we're really pushing the field forward, really glimpsing at the things that are the most confusing, complicated, and unknown, it's a success to make a mistake if it advances sort of our conceptual understanding. It gets us a little bit closer to the truth. And so this is, science works in fits and starts in ways that have been, I'd say, exploited in some political corners to then sort of cast doubt or aspersions on a lot of scientists. And I'm not excluding, there are scientists that cook the books in ways that actually turn progress backwards. I'm not excusing that or saying that we shouldn't stop that or work to stop that. But for the vast majority of science, that's not what's happening. It's advancing, not in the kind of clean textbook way, but in a very messy way that it needs that space to breathe and grow. And it's really a shame to see it under attack in so many political corners.
The great example, recent, is the beginning of COVID, when a bunch of scientists got together and they were on Slack or whatever, and they were worried about the origin of the virus before they had any data. Right. And then as they got data, they said, oh, this is clearly natural. We don't have to worry. But that's been used against them, that they hid something, right? Which is totally... Because then you would say that all of science, you hide something because you're always changing according to your data, right?
That's right. And so, 160 years on, would we throw Mendel in jail or cut science funding because Mendel the numbers seem a little too good to be true? No, we have genetics has been, let's just take the Human Genome Project. The small investments there in terms of the payoffs, in terms of health, medicine, the economy, biotech, national security, all of the above, this has been like home run after home run, and yet, under sort of this view, I think this is, I mean, to me, this is sort of the important moment of the, or one of the reasons to lift up
Mm,
this paper as well, is to try to put this into a bigger lens, right? To the, like, 100 plus years, and what is the value of the work, and how easy it is to attack. Like, you could make credible attacks that the numbers weren't exactly right, or that he wasn't interested in inheritance. I mean, nonsensical attacks, which get all of this traction, whereas the actual work, the years that Mendel put into this, right? And there's, this is a great historical record of this, because it continues to be such a revolutionary success in genetics and in biology. But as he describes, sort of his doubt, or the controversy that was stirring up even in his time, or all of the work,
yeah, yeah,
to make these like that, we've really pushed that into the background. And then anyone who sort of launches a half-baked attack can get into the headlines, can get into the social media, can have a following, et cetera, et cetera. And so, it's striking to see that imbalance. Why do we get up and once a month get on here to kick around some science? It's partially this, because we've had the privilege to see how science works across our careers, to engage in it, to really put our shoulder into it, and I don't know that, as imperfect as our sort of communication is, we have to say it, and not just us, everyone, right? And I think this is part of the way that we try to rebalance the books here on what science is versus how it's attacked, and the value propositions that are involved in that.
well, problem is, Nels, is that scientists do science, and they're the ones who see all this, and what the public gets is just the final positive story, right?
Right. Yeah.
They
Yeah.
don't get
Yep.
all the fits and starts, as you say. And so
Yeah.
that was one of the things we wanted to do, was try and convey that here
Mhm.
on this little TWiEVO and all the other podcasts that we do,
Yep.
and give
Yeah.
backstories about what went wrong and what went right. But
Yeah.
we have to keep doing it and reach more people.
No, absolutely. That's why every time we fire up and folks show up with us, we're not bluffing when we're so happy to have you all here, listening in along for the ride, and learning together, as we go.
Got some other people arriving and comments.
Great.
So
Yeah.
let's see. Christopher Robinson is here. I was flying my glider in Scotland. Cool.
Nice. Yeah.
That's
Those pea
great.
plants that were in evidence today are not too far away in the UK. That big collection for the diversity panel. Yep.
So Steen writes, the NSF Plant Genome Research Program has historically been important for work like this. Not sure whether it will remain an agency priority.
Yep. And my God, and talk about an agency under stress. Not just in the sort of 50% plus proposed budget cuts, but even the illegal, clearly illegal and unconstitutional attacks on the resources that have already been allocated for it, that are being pulled
Yeah.
Awkwardly off, or off just chaotically, from that organization. And then I agree, plant biology is often highly underrated, and certainly makes some sense in biomedical science. A lot of the resources go to things like cancer biology, et cetera. The magical thing about all of science, and hopefully we've given a little bit of a hint of that today, are all of these connections. And so the more we understand about plants, it's not just sort of important to agriculture, it's important for cancer biology. How do transposons mess up genes or transcription factors? We see that in tumors as we see it in seed pods. And so all of this is of the same piece. It all works together to build our knowledge. And so, yeah, it's absolutely devastating, both to see the amount of cuts that are proposed, the illegal shenanigans that are going on here, and then, the front lines of the cuts, which are often things like plant biology, ecology, some of these really important fields that are underappreciated, under-resourced, even in the best of times.
Privyet Veronika from Moscow, where it's warm. And
welcome.
A nightingale season.
Nice. Yeah.
Do you speak any Russian, Nels?
Niet.
Yet.
You?
Yet. Just a few words I've picked up
Okay.
from Veronica, like Privyet, hi, and Polka, goodbye, and a couple of things in between.
Oh, yep.
Wouldn't get me much. Endroot says, after doing peas, Mendel tried hawkweed, which unfortunately makes clone seeds.
Oops. Yeah, there you go. The peas, this was, chance and or whatever it is, like this was a great, and as you pointed out, Vincent, sort of the single copy genes at play. That's also not the case for many,
Yeah.
like, some of the ploidy of, if you picked strawberries, for example, I don't think any of this, we would be telling a very different story.
Philip is in Wales. Welcome, Philip.
Welcome.
Steen says pea is an annual. There are apparently some interesting perennial legumes, clovers and things, but apparently not temperate adapted. Okay.
I see. Yeah. Which would have been, you kind of, that's another sort of historical science point, right? Like you end up studying the things that you're surrounded by.
Yep.
It's great.
Konami Kim says hi. Hello.
Welcome.
Tom says, what strikes me is the different or diverse mechanisms that resulted in these alleles, right, that we kind of just summarized. And it probably happens everywhere right now. It's not just unique to pea.
Oh, absolutely. And this is, one of the drivers of evolutionary biology is this getting to the same outcome through a different route, or independent evolution, convergent evolution. That signal, really interesting and exciting. Sort of, the other side of the coin. So in the last 50 years, the model system genetics era, where things like yeast, flies, worms, mice, we sort of assumed that everything from our common ancestor that's shared, the genetic changes with our common ancestor, those are the important things that have happened. As we've gotten deeper into the evolutionary weeds, what we see is there's like a lot of convergence, but that can be a signal of importance, but with different outcomes. And there's, a lot of meaning in that, which is one of the things that keeps me in the business. Yeah.
Steen says, shout out to SUP figure 39, where they again use virus vector knockdown to validate the organ size locus.
Yeah, that's right.
Figure 39, so many SUP figures.
Yeah, this is sort of the modern scientific publishing in high-profile venues, right? So the amount of data here that they're weed whacking through or presenting.
In fact, Steen says the main article is a kind of summary of the book-length supplement. And also nice that the 33-page referee report and response is available. That's cool.
Yeah, agreed. And open access as well, which is so you don't have to have a subscription to Nature to get this information.
So FSM, now going back to anti-science, I had an anti-vaxxer saying that because Jenner got lucky with cowpox, that science is wrong.
I don't know, because Mendel got lucky with pea plants. Is genetics wrong? It just
it's not at all.
Follow.
Well, it's not logical at all, right? That's the problem.
Yep.
FSM says, were peas the one we heard about because it worked? The fails were binned?
Yeah, this is great. So, and I don't know. Yeah, was there another Mendel who was messing around with strawberries or something and it didn't happen? This is, and I love this because most of science, most of biology are things we can't see, like the things that didn't work. And then the historical record is sort of the ones that did work. Maybe a corollary of this, I'm trying to mix and match our comments here all the way through to pox viruses. So Mozart had smallpox when he was a kid, at a time when between a third or half of the children infected with smallpox died. So coin flip that Mozart lives through his childhood, goes on to compose all of the music that we now famously listen to. But then I think, getting to this comment, how many other kids in Mozart's era didn't live in that flip of the coin? Because was there a super Mozart in there? And what did her music sound like? We'll never know. But right. And so this is, I think it's important to kind of, give it that, try to put these things into that perspective.
It's a genetic roulette wheel, right?
Yeah.
Miss Martian is from New Mexico. Welcome.
Welcome.
And Joseph, Southern Ontario.
Fantastic.
Thanks for coming here. Now, do you want to do some picks now?
Yeah, let's do some picks. So mine is actually a game. This is actually evolution related here. So rarity in these days. It's called Metazooa. And so we've got the link here, metazooa.com. And so metazoa, that's Greek for animals, the animal kingdom, full of distant relatives. And so this game is to every day, metazoa picks a different mystery animal. You get something like 20 guesses to try to figure out what animal it is. And then you make a guess and it tells you, so wrong guesses are ranked by the taxonomy it's in. So it starts building a tree with your guesses that puts your guess underneath the category of animals it's in. So then you have to guess a different animal that's in that same category or a relative.
Right.
The fun thing about this is so it goes through, all of the kind of Greek here of kingdom, phylum, class, order. It keeps showing you that. And so, we know the animals and you guess the animals by their common names, but it sort of puts it into that ranking. And you start, as you're just guessing animals, it's kind of fun. You start to learn how these animals are related to each other and sort of how their taxonomy is described.
That's cool.
And so, yeah, it's pretty fun. Every day they pick a different animal and you can get hints to kind of narrow it down. But it has sort of the side effect of sort of teaching you taxonomy.
That's great. I love that. Yeah.
Mm-hmm.
That's what really I like about it. It's fun and you learn.
Exactly.
Those are the two key things we need.
There you go. So this one, I started out, I guessed a box turtle, and then I went from there, a rattlesnake, an aardvark, and then it was marching me through the amniotes, the euthia, as we narrowed down the carnivores, and so all stuff that I'm not familiar with. I'm not a taxonomist, but it's really fun to do that. And then I won't give away today's animal in case anyone goes to metazooa.com to check that out. But yeah, fun game. I agree. Sort of. Yeah. How about you, Vincent? What's your science pick of the week?
So I have an interesting opinion in Medscape. Mm-hmm. It is by Arthur Kaplan, who is the head of the Division of Medical Ethics at NYU School of Medicine here in New York. Yeah. And the title is Should Unvaccinated Patients Get Organ Transplant? So basically, this is about a 12-year-old girl she's at Children's Hospital. It's in Cincinnati. Mm-hmm. She wants to get on the heart transplant list. And the hospital says you need to be vaccinated against flu and COVID to be on the list. And the parents said no.
Wow.
And so this is a huge backlash, in part because the young girl's mother is related by marriage to Vice President J.D. Vance. Okay, one of his siblings.
I see.
So it gets a little more publicity than possible. And so the girl has been ill since she was 4. They knew she was going to need a heart transplant. But so they picked Cincinnati because they thought it was good. And the ethicist here says, the hospital is just trying to make transplants efficient because if you don't get vaccinated, when you're transplanted, you're immunosuppressed, you're going to get infected, you're going to die. And it's not really a good outcome for you. So get vaccinated. And the hospital, the parents say it's against our religious beliefs. And so the ethicist says, it's hard to know what these religious beliefs are because there are no major religions that oppose vaccinations. All major religions that he's familiar with support vaccination as good for the individual to preserve their health and good for the community. And in fact, yes, there's only one religion that prevents vaccination. So this is a nonsense excuse. It will protect your child. And now your child is not going to get potentially a life-saving heart transplant. This is a very, very tough decision, which shows the effect of anti-vaccine sentiment.
No, exactly. And for me, maybe like stepping back, kind of a tragedy of the commons scenario. So it's like, think about the generosity that goes into a heart transplant, right? Where someone says, I want to be a donor. I want to help out. And, the sort of tragedy in that part of the story, where if someone is in a car accident or motorcycle, whatever it is, right, and then that heart becomes available, but that generosity or that gift, which I think there's an obligation to meet that then with also some
Yeah.
generosity as well. And so if it's like, well, I want that heart, but I don't want to get vaccinated, um, not only as Kaplan is pointing out here, by the way, an old Minnesota connection. I think he was for many years at the University of Minnesota before he migrated east, and followed his career with interest for a long time. But so in a couple of levels. So one is, the likelihood that the patient, as someone immunocompromised, might then that heart might not last very long in the new patient, but also, everyone else who's in the hospital, who's a lot of people who are immunocompromised, and just the spread of disease. So the tragedy of the commons is, well, if I don't get the vaccine and everyone makes that selfish decision, then we're allowing the viruses to
Yeah.
run ramshot, right? And so on that one hand of saying, well, I want the generosity of the transplant, but I don't want to extend that back by sort of doing my part to get to herd immunity. And I think as you get deeper into the weeds, so the sort of religious traditions, or the rare ones, I don't know, like for with measles, the Mennonites, which is part of the story of the current outbreak that started in Texas. That's I kind of am hesitant to, like, in that case where I think there's some real religious beliefs here, but at numbers that are so small that if the Mennonites don't get vaccinated, but we're at herd or well above herd immunity where we've been for measles for decades or longer, there's room for sort of real deep held beliefs where we're still past herd immunity. But in the cases where it's this kind of cooking the books, inventing new stuff, buying into sort of post hoc justifications for anti-vax based on, I
Yeah.
think,
Yeah.
pretty thin convictions and a lack of generosity. That's where I see that disconnect between sort of the J.D. Vance view of Christianity in the world and
Hmm.
what the Pope is saying in terms of actually, these sort of modern strains of religious hypocrisy are starting to sound pretty weak. Yep.
So he writes, the hospital is trying to be responsible and say they don't want her to die if they transplant her. The way to maximize her chance of doing well is to give her vaccines. This makes good sense to me. It also makes good sense to say that we don't want to waste a scarce heart on someone. So 12-year-old heart is very hard to get,
Yeah,
harder than for adults.
of
Waste a
course.
scarce heart on someone who's likely to get sick when other people are waiting in the queue, other kids not related to J.D. Vance who are vaccinated and will do better. From a moral point of view, we want to steward the supply of scarce hearts and make sure they go to the child who has the best chance at life. So I think he says, I think it's right to say vaccination is something the hospital really wants to see. It's medically indicated and for anybody on a wait list for a transplant. And it's not the same and shouldn't be mixed up with fights about mandatory vaccination to get
Yeah.
into school.
Yeah. Yep. Yeah, I'm with Kaplan on this one. Yep.
But these medical decisions are tough. Many parents bring their kids in and then they say, we don't want any anything done because our religion prevents it. So then the hospital gets in a fight. So why did you bring the child in the first place then? What did you
Yeah.
think? They were going to wave a wand and the child would get better? No, they have to do things.
Yeah. Yep. No, and just stepping back again, there's just the stress on academic medical centers on coming out of the pandemic when doctors were risking their lives in the early days before the
Yeah,
SARS-2
yeah.
vaccine. I think that's another place where just more and more pressure, the thing that our expectations as patients, as communities, versus the resources or the lack of resources, as we strip resources from biomedical science. This in both direct and indirect ways puts more stress on the system of medical providers, nurses, doctors, pharmacists, everyone involved in this. And then that mismatch becomes more glaring. And so, yeah, here we are.
And Elizabeth says, imagine you trusting a doctor to do a heart transplant, but not to give a vaccine. The disconnect is insane. Totally
Yeah,
insane. Yeah.
great point. Yep. Yep. Well
Should we
said.
wrap up TWiEVO 114, Nels?
Let's do that. We've this has been fun. We've kind of went back 160 years up into a paper published last month and drawn some lines in between. I really enjoyed it. Thank you everyone for coming and commenting and being in the game, in the mix with us. And we'll be back 115 next
about a month. Towards the end of June, we'll be back.
We'll be back. Thanks,
Nels, you're going to ASV?
Ooh, not going to make it, unfortunately, this year. I've been out on the road, just back from Chicago. So one of the places where I'm seeing some glimmers of hope for science are in some of the private or nonprofit foundations that fund scientists' early careers, postdoc fellowships, and seeing some of the resources dedicated there and being part of, so I'm on a review board for the Jane Coffin Childs Foundation. They do about 30 postdoc fellowships a year, 3-year funding for people who've just got their PhD and are sort of building their own independent research programs. And so I was involved in that process. So anyway, a lot of traveling and fortunately not at ASV. You'll be there though, Vincent?
We'll be doing a TWiEVO, yeah.
Awesome. That's a great meeting. Great society too.
Yeah. That's TWiEVO 114. You can find the show notes at microbe.tv/TWiEVO. If you have any questions or comments, you want to send in a pic, TWiEVO@microbe.tv. And please consider supporting our work, microbe.tv/contribute. I want to thank our moderators today, Barb Mack, Vanity Nutrition, and Tom Steinberg. Really appreciate your helping out, and I want to thank everyone who's here. Nels Eldey is at cellvolution.org and on Blue Sky, L. Earlybird. Thanks, Nels.
Thank you, Vincent. Great to be together.
I'm Vincent Racaniello. You can find me at microbe.tv. The music you hear on Trampled by Turtles on TWiEVO is by trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, be curious. Thanks, folks.